Okay, welcome, my dear friends. Regarding of CPGET free online coaching for 2025 and all competitive exams in the field of plant sciences, today's our eminent resource person, Associate Professor Dr. Tirunahari Ugandhar sir, working in Kakatiya Government College, Hanmakonda, Warangal. Sir has huge experience in the field of molecular biology, genetics, plant biotechnology, tissue culture. Sir is going to explain today's our class in molecular biology, that is nucleic acids. It is the most important topic for all competitive exams regarding of life sciences, CPGET, CSIR net, UGC net, and AP set and TG set, and other competitive exams. Not only botany but also zoology and biotechnology, microbiology. So, my dear friends, utilize this wonderful opportunity. Sir is going to explain in wonderful manner, beautiful with beautiful slides. So, sir, please welcome and start your session, sir.
So it is a so now the nucleic acids. So what is the meaning of the nucleic acid? Acid means is also nucleic means in the nucleus, which has in separate part of the cell that is called a nucleus. In the center part, the nucleus, which have 4 parts are there. In the last class, completed the nucleus structure. Presently, in the nucleoplasm, which have in a separate particles that is called a nucleic acids. So these nucleic acids, which can be majorly in the biological macromolecules, micro or macro, it is also macromolecules that store and transmit genetic information in living organisms. In the living organisms, they are essential for the creation of proteins and inheritance of the traits. Creation of proteins and also called inheritance. Inheritance means it can carry away parental characters or genetical characters from the parent to daughter generation. So this part, the types of the nucleic acid is 2 types. Deoxyribonucleic acid, DNA, found in the nucleus of cells, contains the genetic blueprint for the development and function of living organism. First one. Second one, also called ribonucleic acids, RNA. The DNA structure, double standard helix basis adenine, thymine, cytosine, and guanine. But RNA, it can found in the nucleus and cytoplasm. It can help in the protein synthesis. The structure cytosine and guanine. This is also major 2 type of the nucleic acids, which is important for the living organisms, which can majorly, which can in performing the first one, the inheritance character. The second one is also protein synthesis of this. So these components of the nucleic acid, each nucleotide building block of the nucleic acid contains sugar, azúcar, deoxy, sugar, phosphates, nitrogen bases. The sugars, the deoxyribose in DNA, ribose in RNA, a phosphate groups. Only ribose means single standard. Sugar, deoxy means deoxy sugar is also 2 standards. A nitrogen base, a function of the nucleic acid is the store genetic information, transfer genetic information, direct the synthesis of proteins. It pass hereditary and trails from one generation to the other generation. That is called a major part. So what is in the DNA, the nucleic acids, the first, the nucleic acid which is called a DNA, The second one is called a RNA. In the present, the structure of the DNA. So what is the DNA? Who invented the DNA? What are the types of the DNA? What are the function of the DNA? How to replicate the new DNAs? These are all of the topics are it can study it in this topic of the PPT. Then it is also where is the DNA present in the cell? The cell which have separate or a brain of the cell, that part is also called a nucleus. In the nucleus, the nuclear membrane, nucleoplasm, nucleolus, and chromatin bodies. These nucleoplasm, which have separate acid nature, that is called a nucleic acids, which has a in the center part of the nucleus. You have seen this part, that every cell which is also have separate nucleic acids or part is present, that in the nucleoplasm. So which is also the in the nucleoplasm, that is the DNA structures. So the DNA means also the deoxyribonucleic acids. It is a genetical material. First one. Second one is also RNA. Then so this DNA structure is this part. The DNA is which is a part of the chromosome, a part that chromosome which can be separated or divided into the chromonym of elements. First, chromosomes are divided into the chromonym of elements, and also called the chromonym threads. Each chromonym threads which have a separate DNA threads or formation. That is called a one part. Then what is a special about DNA? What are the special quality? RNA, DNA, what is a both are important, but the DNA is a more genetical part, and also RNA is also genetical part in virus. But DNA is also all organisms have a separate genetical part which can give us a life. DNA is a one of the most boring macromolecule in imaginable substances. It's a made of the only four building blocks and has a perfect monotonous structure. Then it in a just site where it does not catalyze the reactions for a build of the so build cells of organism, so that so good about or what are the about of DNA. So this answer is a in DNA ability to store the copy of information, ability to store of the genetic information. That is also first point. That then what are the DNAs are the DNA present in the so first one in human cells, and also got a plant cells. Then human cells have also chromosome in plant cells also same. The chromosome which have centromere either sides are chromatids. These chromatids are which can be divided into the chromonym filaments. These chromonym filaments are directly to continuation of the a single or a large number of the threads-like structure, that is called DNA. It is also double helix, also sugar phosphates and backbone. Backbone part is there. So this is also always the DNA. It can be a movement to condition of the life. Life means it can breathing, and also called breathing is also respiration. Take the oxygen and give out the carbon dioxide in daily. That person is also live means it can total function is based on the DNA activity, DNA function. This is also movement in always in the nucleoplasm of the nucleus of cell. So these are the two views of the double helix models. The first model is which can separating of the all molecules, only single DNA threads are there. Here is always all atoms are it can be attached to the, so this DNA is almost always wrapped around the proteins. This is also called the chromatin. It is not this what's found the cells. This but here is a timeline. Who invented the DNA? So up to 18th century to 1952, between that period, the DNA is reporting the scientist. In 18th century, a few measure nucleic acid is reported by this first scientist. In 18 means the measure is it can be the genetical and the new organisms are originated by pre-existing organism. Means what is a pre means before or post organism which can carries a inheritance character. That carries inheritance character is storage in the nucleic acid that is reported in the Fisher F. Fisher Mesher scientist. Second, 1928 M.B. Griffith scientist. So this is also Friedrich Griffith transformation of principles. Transformation means one genetical materials are transferred to the other through the bacterial cells. Every MC cleared and MC CAR-T transforming principles in DNA. Then also called the Griffith scientist who was a Hershey-Chas blender experiments all type of the reported in the up to 18th century. 18th century to 1952, this total report of the DNA, it can be completed that. So the next part is also, so what are the quick DNA organization into the chromosomes which are found within the nucleus of cells. Then a gene is a segment of the DNA. Then and a chromosome that codes for the special protein and thus determine the traits. It is also one character. This deoxyribonucleic acid generally genetical material of the cells. Genes, units of the gene, genetical material that is called codes for a specific trait. It called the nucleic acids. Then DNA is made of the repeating molecules called the nucleotides. That is also major different. The DNA nucleotides are there. It is also phosphates, sugars, and nitrogen bases. These nitrogen bases are adenine, guanine, cytosine, thymine is also present. This between then the phosphates and nitrogen bases and sugars are there. The sugars are ribose sugars and deoxyribose sugars are majorly differentiation of the RNA and DNA. That is also next point. Then a history of DNA. Then, so before scientists, before Watson and Crick period, then Chargaff, Friedrich Mischer, and all scientists are reporting the genetical material is called protein or DNA, which is a correct that can have separate so many and differentiations. So this differentiation is a proof by our pioneers of the DNA reporting scientist that is called a Watson Crick. A discovery of a DNA double helix. Friedrich Griffith discovery that factor in diseased bacteria can transform harmless bacteria into dead bacteria. In 1928, that is called a first pioneer of the before DNA reporting, that is Friedrich Griffith scientist report. The second point, Rosalind Franklin X-ray photo of DNA in 1952. So the DNA structure is also reported before Watson Crick. Then after Watson and Crick described the DNA molecules from Franklin X-ray 1953. The Franklin reporting is correct, explained to prepare a separate paper, that publication paper which are published in the 1953. This paper is published here, this one, the discovery of DNA structure by X-ray diffractions and technology. So this the X-ray diffraction base to study of the structure of the DNA is reported. So this DNA X-ray diffraction pattern produced by the DNA fibers, Rosalind Franklin and so Maurice Wilkins and Franklin's, both are first before Watson Crick invitations. That it can reported that after this paper is published in the DNA genetical material is also that can important for the cell and also living organism. That report was published in this paper. So this is also molecular structure of the nucleic acid nature. In the nature, that first publication papers published papers in April 25th, 1953. That year is it can starting of the only simple structure, which is also all scientists are acceptable structure of the DNA, the landmark publications. which is also nature that can, after 1953, the DNA is completely reported or structurally, functionally, the complete explained by Watson Crick only. That is also when next part, then Watson Crick is also pioneers of the DNA. So the Watson Crick model is a proposal for the present days to following. The DNA is a double helix. In the 1951, James Watson learners about X-ray diffraction pattern projected by DNA, knowledge of the chemical structure of the nucleotide, deoxyribosugar, phosphates, and nitrogen bases. These three components are is called the nucleotides. Erwin Chargaff experimentals demonstrated that the ratio of adenine and thymine is 1:1, and the ratio of a guanine and cytine is 1:1. Equally, purines and pyramids. One purine, one pyramids are take hydrogen bonds to joined to each other of adenine, thymine, guanine, cytosine, always bonds to each other to formation of the double helical model of the DNA. In 1953, James Watson and Francis Crick proposed their double helix model of DNA structure in the 1953. This paper is published in 1953, April 25th. 1953, that report is present or published in the Nature publications. Nature publishers. It is a great role. Then Watson and Crick proposed that DNA had a specific pairing between the nitrogen bases, adenine, thymine, guanine, cytosine. That is also pair. So DNA was made of the two long strands of nucleotides, arrangement of the specific way called the complementary rule, two arranges rule, and the DNA made by the two long strands. That complementary rule is one DNA thread, the other DNA thread is linear model. These two DNA linear model threads is joined by the adenine, thymine, and cytosine, guanine in hydrogen bonds. That is important. So this is the structure of the DNA here. So, but this is also proposed the how can DNA store and copy information. So the guanine, cytosine, guanine, all is also joined to each other form. So this is also DNA replication process. Oh, this complementary base, complementary means one thread or two threads are, it can join it between two threads by hydrogen bonds of a adenine, thymine, and guanine, cytosine. Complementary base pairing allows each strands of DNA to serve as a template for DNA replication process. This is also one part. The DNA replication is simple type of the message. But DNA is made of the two long chains of the nucleic acid joined by hydrogen bonds. So this point, hydrogen bonds are a nucleotide. So this is also called a adenine, but it is thymine. So it is thymine, also adenine, guanine, cytosine. It is a guanine and cytosine are complementary as are adenine and thymine. So this is also parts. Then double helix models, the nitrogen bases, which are a adenine, thymine, guanine, or cytosine, long and legs of the in the ladder shape. Then between it is also phosphates and sugar backbones are there. Phosphates and sugars are backbones. So this is also called a DNA double helical structure. Guanine, cytosine, triple hydrogen bonds. Three hydrogen bonds are take fixed there. Adenine, thymine always double hydrogen bonds fixed there. So then either side of the nitrogen bases, sugars are there. Sugar. So sugars either side phosphates. First you that phosphates, sugars is a one thread in the after sugars, then nitrogen base are there. So these nitrogen base after then also got a phosphates and nitrogens. This is also links between the two type of the complementary helical structure of DNA. That is structurally, the nitrogen bases, it is also purines and pyramids are both are different. The two type of the purines and the two type of the pyramids are not same to bond takes place. One purine is one pyramids to take a bond. But adenine is always bonds to the adenine is always bonds to the thymine. Guanine is always bonds to the cytosine. This is also a complementary. And the Chargaff rules, one purine, one pyramids are take bonds. Then that bond is also important for the guanine, cytosine is also tri bonds, three hydrogen bonds. Then thymine and adenine is also double hydrogen bonds. So this bond is always tightly to pack. The DNA is functionally active. So during the replication process, the hydrogen bonds are liberates to separation of the two DNA threads. Each DNA threads metamorphosis into the replicates to two DNAs. One DNA is a parental, other DNA is a new or daughter DNA. Means one self-replication process, only single DNA to formation of the two DNAs. In the same way, the cell is undergoes mitotically to form one cell, two cells or four cells in the same way. So this type of the functions. The nitrogen bases are adenine, thymine. It is also called a Chargaff rules. The Chargaff rule is important rule to the complementary helical structure of the DNAs. Then adenine must pair with thymine. It is a one part. One part of the first rule of the Chargaff. Second one, guanine must pair with a cytosine. Then their amounts in a given DNA molecule will be about the same. That is also one. The amount of the each and purine or pyramid is equally same. Thymine, adenine, guanine, cytosine. It is a Chargaff rule is important for to understanding the DNA structure. This DNA structure, this base, the Chargaff rules are taken to prepare separate method, separate structure by Watson Crick scientist. So these are base pairs. Hydrogen bonds, guanine, cytosine, three hydrogen bonds, thymine, adenine, two hydrogen bonds. That is also base pairs. Pairs or bases means also nitrogen bases. The nitrogen base look like guanine, also purine and a pyramid, which is always takes a bonds. That bond is also got hydrogen bonds. The nucleic acid, the principal information molecule in the cell, all the gene codes are carried out to the nucleic acids. So these nucleic acids are a linear of a nucleotide structures. Then it is also called, that nucleotides, adenine, cytosine, guanine, thymine, or uracil. Then uracil, thymine, also in the space of the uracil. Then in RNA, the nucleotide is composed of the three components, nitrogenous bases, pentose sugars, phosphates. Nitrogenous bases, it is also same, repetition, purines and pyramids. Then also called a DNA building blocks. It is sugars, bases. Then, so this is also called all arrangement of the purines and pyramid structures. It is also genetical variety, diversity, the different arrangements of the nucleotides in a nucleic acids. DNA provides the key to the diversity among living organisms. All type of the organisms are not in same, but the color and morphologically, physiologically also difference based on the arrangement of the nucleotides in nucleic acids. That is also genetic diversity. Arrangement of the nucleotides are not same in one plant or other plants within the species, within the genera. That is also difference. It can exhibit it to the different characters of the human beings, different character, different thinking powers, also difference. So, but the code of the life, the code of the chromosome is the specific order that basis occurs. Adenine, thymine, cytosine, guanine. It is also pair, one code of the pairs. This coding arrangement is not in similar except to and twins. The twins are just close qualities, not more than But in the other, without twins, definitely the code is also the different nucleotides arrangements. The arrangements of the nucleotides are dissimilar in the non-twins. The twins are also similar. That is us. So this DNA is wrapped highly around and histones and coiled tightly to form a chromosome structures. So this linear polymerization of the nucleotides means the nucleic acids are formed of the nucleotide polymers means also the nucleic acids are polymerized together by phosphodiester bonds was condensation reactions. The phosphodiester bond is formed between the phosphodiesters also got hydrogen bonds are it can formation to between these two. The polymerization of the nucleotides here is also there. Then polymerization of the nucleotides are nucleotides. The nucleotides formed of purine or pyrimidine or pyrimidines bases like to the phosphorylated sugars also got a nucleotide backbone. The bases are the link to the pentose sugars to form the nucleoside. Pentose here is also differentiation of the nucleotide and the nucleoside here. The nucleotide is phosphorylated sugars. The nucleoside is pentose sugars to form. The nucleotides contains a one phosphate group linked to the other five carbons of the nucleosides. The nucleotide is called nucleoside plus phosphate groups. Phosphate groups only without phosphate group is called nucleoside. With the phosphate group is called a nucleotide. Means phosphate means sugars and nitrogen bases is called a nucleoside. Sugars, phosphates, nitrogen bases. Three is called a nucleotides. That is also differentiations of the nucleotides and the nucleosides. Then the complementary base pairing. It is the most important structural feature of the nucleic acids. Complementary. It contains the bases of one polynucleotide chain, nucleotide polymer, with a complementary bases of other chains. So this is the complementary bases are bonded together by many double hydrogen bonds are between adenine and thymine and uracil RNA adenine thymine or adenine uracil. Adenine uracil adenine only thymine is different. In the DNA is thymine, also called RNA is a uracil. Triple hydrogen bonds between guanine and cytosine in both DNA or RNA. Guanine and also called cytosines are in the similar to the RNA and DNA, but DNA is only adenine and RNA is also adenine and uracil. Then DNA is adenine thymine. That is also just differentiation. Thymine is also instead of the thymine, which uracil is also present. But here is also base pairing parts. Base pairing parts. But significance of the complementary base pairs. What are the complementary base means? The two threads of the DNA base pairing, each strands of DNA can act as template to direct the synthesis of other strands, similar to its complementary one. Complementary means right and left hand, the two type of the DNA threads which have same type of the structure means one thread to under the replicate two new DNA threads formation. The second thread is also called other two DNA threads formation. Thus, the nucleic acids are uniquely capable of directing their own self-replication. Means that is called a rule of the complementary base pairing. The information carried by DNA and RNA direct the synthesis of specific proteins which control most cellular activity. So this is a important of the significance of a complementary base pairing, how to pair the two threads of DNA. So this is also important of the one thread, other thread, both are joined by adenine, also thymine, adenine, guanine, cytosine, adenine, thymine. That is So these are then also got the second DNA replication. So how to replicate the DNA? Means the DNA is the old DNA to develop a new DNA. Means the old DNA, the new DNA is originated from pre-existing DNA. The DNA must be copied. The DNA molecules are produced two identical new complementary strands following the rules of the base pairs. So the each strands of the original DNA serves as template for the new strands. It is also the first pair is also taken separation of the DNA threads. Second one is a total separation. Third one is one is a blue color, other is also red color. The two DNA threads are new formation in the third figure, which is also DNA. The DNA replication is which divided the semi-conservative model. Watson and Crick showed the two strands of the separate and each functional as the first synthesis of the new complementary strands. So this is also two complementary strands of DNA. These two complementary strands, each complementary strands DNA, the new DNA, old DNA templates. But here is a second complementary trait or complementary DNA formation of the blue color and red color. That is got a semi. Semi means off. The off complementary DNA to formation of the remaining of other of complementary DNA to form the other new. That is also two DNAs or two DNA formation from the single DNA. That is a essential feature of the BDNA. So how many type of the DNAs are there? Then the 5 type of the DNAs are present that can base the right twisted double standard of helix is called a BDNA, and the parallel base on the inside perpendicular axis, uniform diameter, major and minor grooves are present. The complementary base pairs that is called a BDNA. Structurally, purine and adenine and guanine pair best with pyramid and pyramids, thymine and cytosine. Thus, a pair with thymine and guanine pairs with cytosine also explaining the Chargaff rails. So this is also important of the BDNA types of the. Then Y-DNA enveloped in the genetical material but not RNA. Because RNA but not DNA is prone to based catalyzed hydrolysis. Catalyzed hydrolysis. This is also the BDNA means also biologically dominant. BDNA, right-handed double helix structures. It plans for base pairs or perpendicular to the helix axis. Perpendicular to the helix axis. Helix axis passes through the base pairs and hence BDNA has no internal spaces. No internal space. That is BDNA special character. Then BDN has a wide and deep major grooves and a narrow and deep minor grooves. Then the major groups are, it is also deep major groups are, it is important of the DNAs. That is character of the characteristic features of the BDNA form. The BDNA is majorly important of the biological organism. DNAs. So this right-handed double helix model plans of base pairs, which is a important of the organisms. And then the DNA conformation, BDNA, ADNA, ZDNA, and also called DDNA. This is also 4 types of the DNA, sir. It can come. Then ADNA and DDNA. What is the differentiation? A and B DNAs. Here is there. Both form the and a double helix. Here is a BDNA helix has a larger pitch and hence a smaller width than that of A. This is also A. A is also wide, more wide is present. In BDNA, the helix axis passes through the base pairs and hence BDNA has no internal spaces. Here is a BDNA is no internal space, but is also in the whereas that ADN diameter along with helix 6 diameter engrams. But the plans of the base pairs BDNA are nearly perpendicular to the axis, where in ADNA they are inclined from this. Therefore, BDNA has a wide and deep major grooves. Wide and major grooves are present in the BDNA, but not A. That is also differentiation of the in structurally BDNA ADNA formation. Here is a ZDNA. The BDNA form a right-handed double helix in which the preparing unit is a nucleus, where ZDNA form a left-handed. That is also ZDNA, you know that here is also left-handed, but BDNA is also got a right-handed. Then also unit in A dinucleated, the ZDNA helix has a larger pitch and is therefore the narrow that has BDNA. BDNA has a wide and deep major groups. Wide BDNA wide and major groups are present. ZDNA has a narrow and deep minor. That is also A first A and BDNA. Here is also Z and BDNA. Both are different regions are. So this all four is there. So first one ADNA, BDNA, cDNA, ZDNA. So these are all DNAs are structurally also different. The minor groups or major groups are also different here. All. So that is also in the structurally, but here is also in the form of DNA, ADNA, B, C, D, and E DNA or ZDNA. All of the total figures are here that. Then coiling, pitch, base pairs, and helical diameter, axial rise per sugar passed by backbones, title of the base pairs. Here is all differentiation. This differentiation is the total how many characters are based. The 7 major characters are based to differentiations. That is called a general characteristics of the several forms of the DNA. But here legal twist base. A right 11, right B right 10, Z left 12. That is also a basic twist are there. That is carried. So here is also BDNA majorly. BDNA, ADNA, and ZDNA are differentiations that can. So this is also comparison. So how do comparison of the BDNA and ZDNA means here the majorly it is also helix since right B DNA is called B form, right-handed also helix A form, but only Z is also left. B and A is also same. Z is also left-handed. In the base pair per turning, here is a B form is also 10, A form is also 11, Z form is also 12 turning points. That vertical rise B base pairs. B form is also 3.4, A form is also 2.56, Z form is also 3.7 angstrom. Then rotation per base pair is equal to B form plus 36 rotations. A form is also plus 33. Z form is also 30 minus 30 degrees. Then helical diameter is also B form is also minus 19. Then right A form is also 23. Z form is also 8 angstrom. This is also comparison of all character features of the 3 major DNA types. Here is a total DNAs are how many types are there? What are the differentiations? A DNA, B DNA, Z DNA, C DNA, mitochondrial DNA, chlorophyll DNA, plasmid DNA, satellite DNA. That is a total how many 8 type of the DNAs are there. So these 8 DNAs, how to comparison, structure and also location and also key features also functions. So these all type of the qualities are total characters, total function base to prepare this table. ADNA right-handed, BDNA also called right-handed, ZDNA also got a zigzag backbones. cDNA means complement, single stranded synthetic form of RNA. Mitochondrial DNA, circular double stranded. Then chloroplast DNA, circular double stranded. Plasmid is small circular satellite DNA, repeated non-coding. But second band location. ADNA is a dehydrated sample DNA RNA hybrids. BDNA, most cells, physiological conditions. JDNA, regions with alternating purines and pyramids. cDNA is created in labs. C, created in the labs, it reverse transcription process to create artificial DNA formation, that is immersion plants. Chloroplast DNA, only plants and algae. Then plasmid DNA, only bacteria and yeast. Then satellite DNA, centrosomes and also got telosomes. But next, aDNA, how many and turnings are there? What are the function of that there? Then 11 BP turning base title rarely are found in the living cells. All living cells. But BDNA, 10 BP turns bases, perpendicular most common DNA form. BDNA is the most common forms. ZDNA, zigzag DNA, it can maybe regulate gene expression. During the gene expression, the DNA structurally, it can be changed. That is called zigzag DNA. cDNA means it is also called a created DNA. The no introns used in cloning process. Cloning means it can be artificial cloning, means nature cloning. Nature cloning means plasmid. Inserted of the DNA into plasmid to multiplication of the DNA in several copies. That is called a plasmid vector mediated and a gene cloning process. Without vector, that is called a PCR, polymerase chain reactions. So this is also cDNAs and gene expression studies. Gene. Then mitochondrial DNA is inherited from the mother, involved in energy productions. Only ATPs form based chloroplast DNA. Foods are some photosynthesis related proteins. Then plasmid DNA, it carries extra gene used in genetic engineering. Extra genes, and which type of the genes have additional genes. That additional genes may be covering or identified with can use to the gene cloning in futurely, that can base to the identification of the plasmid DNA structure. Then satellite DNA structure role helps in a chromosome stability. So this is also total. Then 8 type of the DNAs are major important for it can be classified based on the all type of the cell in the nucleus, out of the nucleus. Out of the nucleus means mitochondrial DNA, chloroplast DNA, and also plasmid DNA, satellite DNA. That is, inside of the nucleus is aDNA, bDNA, zDNA. Artificial means cDNA. That is also the total DNAs are here is also completed. But what are the function of the DNA? What are the function? The function and explanation. Genetic information storage means DNA storage the genetic blueprint of all living organisms. Blueprint means model for how to develop, how to formation of the new organism from these parents to idea. That is called a blueprint. Then transmission of heredity means DNA passes genetic traits from parental to offspring during reproduction. That is called a transmission heredity in functionally. Then protein synthesis. The DNA provides the instruction or genes for making protein via mRNA. The protein and mRNA, what are the differentiations or studies in the protein synthesis? Also important function. One of the function of the DNA. Then fourth one, cell division and growth. DNA replicates before cell division. Means S1, S2, G stages. That the before cell division, ensuring each new cell has identical DNA. The old DNA quality character to identically new DNA formation in the mitosis. That is called a one cell division. Then the regulates the cellular activities. Then DNA controls cell functions. by regulating gene expressions. It can control the cell, all cell organelles functions. Then mutation and evolution also important role in the DNA. Changes in DNA mutations lead to genetic variation and drive evolutions. Genetic variation, genetically different, means based on the environmental characters, how to changes of the genetical to formation of the evolution in olden days to present days. That is studies the mutation and evolution, repair and maintenance. DNA as self-repair mechanism to fix errors and maintain stability. Stability means does not change when atmosphere, when environmental conditions, it may be changed in few, not only more. Then formation of the RNA, DNA acts as a template for transcription to produce RNA. That is also 8 important functions performing cell organelle is called a DNA or deoxyribonucleic acids. So this is also complete of the all topics, all headings of the DNA. Now, just one asking the few important filling the blanks. How to prepare the multiple choice. Also, filling the blanks in your future needs. That is also filling the blanks. History of the nucleic acids. The nucleic acid was first discovered by in 1869. Who was that? Friedrich Mischer. That is important of the nucleic acid importing scientist. Friedrich Mischer named the substance. He discovered the pus cells as pus means chemo cells. Pus cells as nuclein. The nuclein word is it is a first word for nucleic acids. The term nucleic acid was coined by in 1889 on Sir Richard Altman scientist. Then the transforming principle in bacteria was demonstrated in 1928 by Frederick Griffith. Frederick Griffith is first scientist to transformation of DNA from in the bacterial cells. Avery, Maitland, and McCarthy showed that DNA is the genetic material in the year 1944. Before 1944, DNA as genetic material or protein as genetic material were confused. After 1944, the DNA only as genetic material, not protein. It can confirm that scientist. So this double helix model of DNA was proposed by, James Watson and Francis. The base pairs rules, adenine, thymine, and guanine cytosine were discovered by Erwin Chargaff, that is major scientist. Then the first complete genome to be sequenced was of the virus is bacteriophage. Then the technique was based by Rosalind Franklin to study DNA structure was X-ray crystallography. That is important. Based on the X-ray crystallography, our pioneers of Watson Crick studied in a double helical structure of DNA. RNA was discovered to have catalytic activity in the form of ribozyme. That is also one care. Then fill in the blank structure of nucleic acids. DNA stands for deoxyribonucleic easy. RNA stands ribonucleic acids. The basic unit of nucleic acid is called nucleotide. Then nucleotide is made up of the sugar phosphates groups. Then the sugar found in a DNA is deoxyribose. The sugar found in RNA, ribosugars. The 4 nitrogen bases in DNA are adenine, thymine, cytosine, and guanine. RNA. Here is also RNA. The RNA is the base thymine is replaced by the uracil. Here is also adenine, thymine, guanine. Same adenine, thymine, cytosine, and guanine, right? In the RNA is the base thymine is replaced in the uracil. Adenine always pairs with the in thymine. cytosine always pairs with guanine. The bond between two nucleotides is called a phosphodiesters. The two strands of DNA run in anti-parallel lines. The two RNA strands are held together by hydrogen bonds, double hydrogen bonds, triple hydrogen bonds. The DNA is described as a double helical structure. Backbone of DNA is made up of alternating phosphate group is called sugars. This is also one fill in the blank DNA replication and its types. So DNA is replication is the process by which DNA makes copy of DNA. Copy of DNA. The DNA replicates occurs during the synthesis phase. S1, S2, that is the enzymes responsible for unwinding DNA double helix is called the DNA polymerase. DNA, the short fragment of the from lagging strands are called the Okazaki. The enzymes that joins Okazaki fragments is called DNA ligase. It is also important. The three theoretical model of DNA replication were conservation, semi-conservation, and dispersive. The three type of the replication: conservation, conservative model, then also called a replication dispersive. The experimental that confirmed the semi-conservation model was performed by Michel Stahi. Michel Stahi was a double helical model of the DNA replication conservative process. So this is also completing of the structure of the for next class. I will take that RNA structure. Okay.
Thank you, sir.
Thank you, sir. Thank you very much.
Excellent class, sir. Complete picture about the molecular structure, components, and types of the DNA and functions of the DNA, and most important bits related to the DNA, components of the DNA, structure, and the functions related to DNA, and the DNA replication, a little bit of the DNA replication, even though the DNA involved as a genetic material, why internal structure, base pairs, and nucleotides, structures, and everything, each and every point you covered in this class, sir. It is very helpful to all the competitive exams related to the life sciences, not only botany, not only zoology, but also molecular biology, microbiology, genetics, biotechnology. So these are the branches of our main botany. So it will help to our aspirants, young aspirants, those who are going to attempt the all competitive exams related to CPGET, CUET, and AP set, UGC, NET, CSIR NET, NTA, CSIR NET, even though ARS NET also. And at the same time, it is also helpful to the young people, those who are going to attempt the related to groups examination also. They are absorbed related to our biology. Means forest department or forest section officer, forest beat officer, and other related our plant science exams. Definitely these each and every point helpful for their better future. Thank you very much, sir. Your wonderful presentation during this 1.5 hour. 1 hour you are presented excellently, sir. Thank you very much.
Okay, sir. Okay, sir. Thank you.